All right, listeners, ready to dive deep into some cutting-edge science. Today, we're going to be exploring
Oh, yeah.
a revolutionary technique that is changing the way we study the brain. It's like getting X-ray vision, but for the brain's activity.
Yeah.
Imagine being able to see
Mm-hmm.
which genes are active
Right.
in different parts of the brain,
Exactly.
all while it's still intact, in 3D.
No slicing and dicing necessary.
And no more treating the brain like
Right,
belly
yeah.
meat, which I imagine is a huge relief for
Oh,
researchers.
absolutely. I mean, traditional methods really limited what we could see.
Yeah.
You lose that crucial spatial context.
It's like trying to understand a city
Mm-hmm.
just by looking at individual buildings.
Yeah, exactly. Like you miss the streets, the traffic flow, how it all connects.
Right. You'd miss the big picture.
TRISCO lets us see the entire cityscape of the brain.
So tell us more about this TRISCO technique.
So it's a bit of a mouthful. It stands for tissue RNA in situ hybridization compatible with clearing.
Catchy.
Yeah, I know, right? But the name, it describes the technique pretty well. It essentially allows us to see which genes are being expressed in a tissue,
Okay.
but in 3D.
And just to remind our listeners,
Yes.
RNA is like the messenger molecule that carries instructions from DNA to build proteins.
Right. It's like the blueprint being read out.
Okay, so TRISCO lets us see which blueprints are being used in each part of the brain.
Precisely. So the first step is to make the brain tissue transparent.
Wait, hold on.
Yeah.
We can actually make brains see-through.
Yeah, it's like clearing a foggy window. They use a special chemical process
Okay.
to remove lipids and pigments.
So we're making the brain
Yeah.
basically invisible?
Well, not quite invisible, but transparent enough that light can pass through it.
That's wild.
Yeah.
So how do we actually see which genes are active?
Right. So once the brain is transparent, they use this really cool technique called in situ hybridization chain reaction,
Wow, that's a mouthful.
or is isHCR for short.
Okay, is hHCR much better?
Or easier.
So what does hHCR do?
Basically, it lets us label specific RNA molecules with fluorescent probes.
Oh, okay, so these probes act like tiny little beacons
Exactly.
highlighting the specific RNA we're interested in.
Mm-hmm.
So it's like attaching a glowing tag to each blueprint.
Yeah. A really, really tiny glowing tag.
And then we can actually see where those blueprints are being used in the brain.
Precisely.
But wait, if the brain is see-through
Yeah.
and the probes are glowing,
Right.
how do we actually capture that information?
Ah, that's where light sheet microscopy comes in. This type of microscopy is perfect for imaging transparent samples
Okay,
because it illuminates the sample with a thin sheet of light,
got it.
allowing you to capture these really high-resolution 3D images.
So basically, we're taking pictures of the glowing blueprints from all different angles,
Yes,
and then using those pictures to create a 3D map of gene expression.
precisely. And this map is incredibly detailed. You can actually zoom in and see
Wow,
individual cells and their RNA expression profiles.
this is incredible.
It's like having a Google Maps for the brain, but instead of streets and buildings, you see genes and cells.
I love that analogy. So we're basically creating a 3D map of which genes are being expressed throughout the entire brain.
And how it changes over time. Like you said, it's like a live feed of the brain's activity.
That's mind-blowing. We're talking about a level of detail we've never had access to before.
And it's opening up so many new avenues for research. I mean, we're just scratching the surface of what's possible. So it's not just static maps either.
Oh.
Yeah, TRISCO actually lets us see the brain in action.
Like in real time.
Yes. Like we can actually see how gene expression changes
Wow.
in response to different things.
That's amazing. So we can see the brain reacting to stimuli.
Exactly. For example, in the paper, they had mice swim for a few minutes.
Okay.
And then they looked at which genes were activated.
So they could actually see which parts of the brain were involved in processing the swimming experience.
Exactly. It's like watching the brain light up
Oh.
in response to a stimulus.
So they saw increased activity
Right.
in areas related to movement and memory.
Yes. And even the stress response.
That's interesting.
It really highlights how interconnected everything is in the brain.
Right. It's not just like one little area
No, not at all.
that's responsible for swimming.
It's a whole network of regions working together.
I see. So TRISCO is giving us this incredible insight into how the brain works on this very detailed level.
It's like a whole new world has opened up.
It sounds like it. What other kinds of things are they exploring with TRISCO?
Well, they've actually used it to study the effects of drugs on the brain.
Oh, wow.
Yeah, for instance, They looked at this drug that's used to treat type 2 diabetes,
Okay.
and they saw how it impacted certain neurons that control appetite.
So they could actually see the drug
Yeah.
interacting with specific cells.
It's like watching a delivery truck drop off its package at the right address.
Wow.
You know?
That's a really good analogy.
Thanks. This kind of detail
Yeah.
could be huge for developing better medications.
It's like taking out the guesswork.
Exactly.
Right, yeah.
And I think that's one of the most exciting aspects of this technology, the potential to really target treatments with much greater precision.
So it's not just about understanding the brain.
No.
It's also about using that understanding
Mm-hmm.
to develop new therapies.
To actually help people.
Yeah. So we've been talking a lot about the mouse brain,
Right.
but can TRISCO be used to study human brains?
Oh, yeah, absolutely.
Okay.
And they've already started doing that.
Have they?
Yeah, they use TRISCO to study human fetal brains.
Wow. So they can see how our brains develop?
Mm-hmm. Even before we're born.
That's incredible.
Yeah. This information is going to be invaluable.
Yeah.
Yeah.
For understanding developmental disorders.
Right, like autism.
Exactly.
But I imagine there are some ethical considerations
Of course.
when studying human brains.
Absolutely.
Yeah.
And the researchers were very clear
Okay.
about the ethical guidelines they followed,
Good.
making sure they had informed consent
Mm-hmm.
and respecting the sensitive nature of this research.
Right, because this is really delicate stuff.
Yes. Yeah.
But the potential benefits are huge.
Yeah. It's like unlocking the
Right.
secrets of the human brain.
Understanding how it develops, how it can go wrong, maybe even how to fix it.
It's really
This
exciting.
is revolutionary stuff.
And this is just the beginning.
Wow.
You know, TRISCO is still a relatively new technique.
Okay.
There's so much more to explore.
Right, we're just scratching the surface.
Exactly.
Of what it can do.
And it's not limited to brains either.
Really?
Yeah, researchers are starting to use TRISCO to study other organs as well,
So we could potentially use this technique.
to see the activity of genes throughout the entire body.
Pretty much. Which is mind-blowing if you think about it.
That is mind-blowing. This is like a whole new era
It is.
of biological research.
It's a really exciting time to be in this field.
It sounds like it. But with any new technology,
Right.
there are always challenges.
Of course. And TRISCO is no exception. So what are some of the challenges?
Well, one of the biggest ones
Yeah.
is the sheer amount of data that TRISCO generates.
Right, because these 3D images
Mhm.
are incredibly detailed.
They are massive. You need a lot of computing power
Yeah.
just to process and store them.
So it's not just about seeing the data.
Right.
It's also about making sense of it.
Exactly. And that's where bioinformatics comes in.
Bioinformatics.
Yeah, it's basically using computer science
Oh,
to
yeah.
analyze biological data.
So we need these really powerful algorithms
Uh-huh.
to help us extract meaningful insights.
Yeah, to find the patterns hidden in all that data.
So this is a real collaborative effort.
It is, between biologists
Mhm.
and computer scientists.
You need both to make this work.
That's really cool.
Yeah.
So besides the data analysis,
Mm-hmm.
are there any other challenges?
Yeah. Another one is the time it takes
Oh, okay.
to actually do the TRISCO process.
So it's not a quick procedure.
No, it can take several days
Wow.
to prepare the tissue
Yeah.
and then do the imaging and everything.
So it requires a lot of patience,
It does.
but I imagine it's worth it,
Oh, absolutely.
especially when you consider
Yeah.
the potential impact this could have
I mean,
on our understanding of the brain.
it's really changing the game.
Yeah.
And researchers are always working
Mm-hmm.
to improve the process.
To make it faster and more efficient.
Exactly.
So it's clear that TRISCO is a game changer,
It is.
despite these challenges.
And it's only going to get better.
Right. We're just at the beginning.
Exact.
This has been an incredible deep dive so far.
Yeah, it's been fun.
I feel like we've learned so much about this amazing technology.
It's definitely cutting edge.
Before we get too ahead of ourselves, though,
Right.
let's take a step back
Okay.
and recap
Mm-hmm.
some of the key takeaways.
Sounds good.
All right, so key takeaways. TRISCO lets us see RNA. And why is that important again?
Well, RNA is like the middleman between our DNA and the proteins that make our cells work.
Okay, right. So by seeing which RNAs are active, we're basically seeing which genes are being switched on and off.
Exactly. It's like a real-time readout of the brain's activity. And TRISCO lets us do this with incredible detail in 3D.
Right. No more flat images. It's like we can zoom in and out of the brain and see how different cell types are connected.
And the applications are huge, like mapping those cell types,
Yeah.
studying neuronal activity, even looking at how drugs affect the brain.
Yeah, we talked about how they even use TRISCO to study human fetal brains.
Right, which could be really important for understanding how the brain develops. And while there are always ethical things to consider, the potential benefits are immense.
Absolutely. It's amazing to think we can see how the brain is wired up before we're even born.
And as TRISCO becomes more refined, we can expect even more groundbreaking applications.
So what's next? What are the big questions researchers are tackling now?
Well, one big challenge is figuring out how to analyze all the data TRISCO generates. These 3D images are super complex.
Right, it's not just about seeing the data, it's about making sense of it.
Exactly. And that's where bioinformatics comes in, using computer science to help us analyze all that biological data.
So we need these powerful algorithms to find the real meaning in all that information.
Yeah, it's a real collaborative effort between biologists and computer scientists.
This has been an incredible deep dive. I feel like we've learned so much about this amazing technology.
It's definitely a game changer.
Any final thoughts for our listeners?
I'd say just stay curious. TRISCO is just one example of the incredible things happening in science right now.
Well said. A huge thanks to our expert for taking us on this journey through TRISCO. And to all our listeners, keep those brains engaged, and we'll catch you on the next deep dive.